We provides information on the dengue virus, includes dengue introduction, signs and symptoms, dengue family, life cycle, diagnosis, treatment and prevention. Dengue is a mosquito-borne infection caused by dengue virus, which is characterized by fever, severe headache, muscle and joint pain, nausea and vomiting, eye pain, and rash. Severe forms of the disease, dengue hemorrhagic fever and dengue shock syndrome, principally affect children. Dengue virus is transmitted by female mosquitoes mainly of the species Aedes aegypti and, to a lesser extent, A. albopictus. The first record of a case of probable dengue fever is in a Chinese medical encyclopedia from the Jin dynasty, which referred to a water poison associated with flying insects. The first recognized dengue epidemics occurred almost simultaneously in Asia, Africa, and North America in the 1780s, shortly after the identification and naming of the disease in 1779. The first confirmed case report dates from 1789 and is by Benjamin Rush, who coined the term breakbone fever because of the symptoms of myalgia and arthralgia. The viral etiology and the transmission by mosquitoes were only deciphered in the 20th century. The socio-economic impact of World War II resulted in increased spread globally. Nowadays, about 2.5 billion people, or 40% of the world's population, live in areas where there is a risk of dengue transmission. Dengue spread to more than 100 countries in Asia, the Pacific, the Americas, Africa, and the Caribbean. Dengue occurs in two forms, dengue fever and dengue hemorrhagic fever. Dengue fever is a severe, flu-like illness that affects infants, young children and adults, but seldom causes death. Dengue hemorrhagic fever is a potentially deadly complication due to plasma leaking, fluid accumulation, respiratory distress, severe bleeding, or organ impairment. Dengue should be suspected when a high fever is accompanied by the following symptoms: sudden onset of high fever, severe headache mostly in the forehead closing parenthesis, pain behind the eyes which worsens with eye movement, body aches and joint pains, nausea or vomiting. Dengue hemorrhagic fever should be suspected when a fever is accompanied by the following symptoms: severe and continuous pain in abdomen, bleeding from the nose, mouth and gums or skin bruising, frequent vomiting with or without blood, black stools, like coal tar, excessive thirst, dry mouth, pale, cold skin, restlessness, or sleepiness. Warning signs occur 3-7 days after the first symptoms in conjunction with a decrease in temperature and include severe abdominal pain, persistent vomiting, and rapid breathing, bleeding gums, fatigue, restlessness and blood and vomit. Dengue virus, a member of the family Flaviviridae and the genus Flavivirus, is a major human pathogen transmitted by mosquitoes. There are 4 ontogenically different serotypes of the virus: Dengue virus 1, Dengue virus 2, Dengue virus 3 and Dengue virus 4. Dengue virus is a 50 nanometers virus enveloped with a lipid membrane. There are 180 identical copies of the envelope E protein attached to the surface of the viral membrane by a short transmembrane segment. The virus has a genome of about 11,000 bases that encodes a single large polyprotein that is subsequently cleaved into several structural and non-structural mature peptides. The 11 kilobits positive sense RNA genome encodes 3 structural proteins. The virus particle consists of an RNA capsid protein complex, surrounded by a bilayer lipid membrane. The proteins present on the surface of the dengue immature virus are the E and M. The mature virus surface, on the other hand, contains E and M proteins. A cell normally uses endosomes to take in large molecules and particles from outside the cell for nourishment. By hijacking this normal cell process, the dengue virus is able to enter a host cell. Once the virus has entered a host cell, The virus penetrates deeper into the cell while still inside the endosome. In the cytoplasm, the nucleocapsid opens to uncoat the viral genome. This process releases the viral RNA into the cytoplasm. The viral RNA then hijacks the host cell's machinery to replicate itself. The virus uses ribosomes on the host's rough endoplasmic reticulum to translate the viral RNA and produce the viral polypeptide. This polypeptide is then cut to form the 10 dengue proteins. The newly synthesized viral RNA is enclosed in the C proteins, forming a nucleocapsid. The nucleocapsid enters the rough endoplasmic reticulum and is enveloped in the endoplasmic reticulum membrane and surrounded by the M and E proteins. This step adds a viral envelope and protective outer layer. Diagnosis of acute or recent dengue infection can be established by testing serum samples during the first 5 days of symptoms or early convalescent phase. Acute infection with dengue virus is confirmed when the virus is isolated from serum or autopsy tissue specimens, or the specific dengue virus genome is identified by RT-PCR from serum or plasma, cerebrospinal fluid, or autopsy tissue specimens during an acute febrile illness. Acute infections can also be laboratory confirmed by identification of dengue viral antigen or RNA in autopsy tissue specimens by immunofluorescence or immunohistochemical analysis, or by seroconversion from negative to positive IgM antibody to dengue or demonstration of a 4-fold or greater increase in IgG antibody titers in paired, acute and convalescent serum specimens. There is no specific medication for treatment of a dengue infection. Persons who think they have dengue should use pain relievers and avoid aspirin. Rest. Drink plenty of fluids. Consult a physician. In late 2015 and early 2016, the first dengue vaccine, Dengvaxia by Sanofi Pasteur, was registered in several countries for use in individuals 9-45 years of age living in endemic areas. At present, the main method to control or prevent the transmission of dengue virus is to combat vector mosquitoes. Thank you.